Hi everyone. I'm Muhammad Saad, is PhD scholar at the Ohio State University. Today I'm presenting the topic redefining the internal recognition of pregnancy in ruminants and emerging inside. That topic is much related to my PhD work as well. I will go to the brief and the general concept behind the internal recognition of pregnancy. Okay, so I'm going to start first with the normal estrus cycle of the cow. As we know, the estrus cycle is of 21 days length. And at the start of estrus cycle, uh, there comes a first wave of follicular wave. And that first follicular wave is usually start with a lot of new small follicle. They go through different stages like recruitment and then selection, and then one of them reach to the dominancy. But not every dominant follicle ovulate. That's what happened with the first wave. Around like 6 or 7 days, you can see a dominant follicle, but it, instead of becoming a mature follicle and to go to the ovulation, that follicle will go to the atresia. And this is because of this corpus luteum, CL, because this corpus luteum is producing a hormone called progesterone, and this will not allow this dominant follicle to get to for the ovulation. So after atresia, there will be a second wave will start, and then the second wave will pass through the same process: recruitment, selection, and then dominance. But then again, because of this corpus luteum, this follicle will not able to go to the further stage of ovulation. Instead, will go to the atresia stage, and then there will be a third stage, a third wave. Then this wave actually will go through the same process: recruitment, selection, and then to the dominancy. But at this stage, the corpus luteum is starting decrease in their size, and the progesterone is already goes down. And then this progesterone block is not more there. That's why this follicle will go to the stage of ovulation. So as we can see, these days between day 14 to 17, they are really important because there is something happening which is causing the luteolysis of this CL, and then that lead to the ovulation of that animal. So why this happened? This happened because of one hormone called prostaglandin PGF2 alpha, which is secreted from the uterus. And you can see in the next slide. Uh, so this cycle will repeat again and again. So here is the hormonal profile of this cycle. If you see at the start of the cycle, when the CL was at immature stage, so the progesterone will start increasing, and then after a couple of days, when the CL become mature, the progesterone is relatively higher and constant through this phase. And then after day 14, progesterone is going down. This is because of this hormone called PGF2 alpha. This hormone is secreted, and then it causes the luteolysis of Cl, corpus luteum, and that's why this progesterone goes down, and then the ovulation happens. So that means if the animal is pregnant, uh, what if the animal is pregnant? So if the animal is pregnant, we need this hormone progesterone, and we need corpus luteum throughout their throughout their gestation length, which is like 9 months in the case of cow. So we want to rescue this corpus luteum. We want to rescue this corpus luteum. We don't want that this corpus luteum go to the luteolysis. In other words, we don't want this PGF2 alpha go to the corpus luteum. So we need to rescue this CL. But how? How this animal will rescue uh, the CL? So for that, we need to actually understand the process of luteolysis, like how this process of luteolysis is happening. So the process of luteolysis, if you see, the endometrium has two type of receptor. First receptor, estradiol receptor, and then the oxytocin receptor. When the estradiol receptor are here, when the estradiol comes and attach with the receptor, they actually increase the receptor of oxytocin. When oxytocin receptor increase and they express with the presence of oxytocin, they actually produce more PGF, PGF alpha. And then if we zoom in this process at a cellular level, you can see the arachidonic acid, basically it is converted by the COX enzyme to PGH2. And then this PGH2 is actually converted by the PGF2 alpha synthase enzyme and then converted to PGF2. And this PGF2 go to the to the ovary and to the CL and cause luteolysis. So if we for that thing, we know that now the day 16 is considered as a critical period for the cow in non-pregnant pregnant cows and also for the pregnant cow as well, because from the ovary there is a dominant follicle which is producing the estradiol, and that estradiol will come to the uterus, and we know the estradiol receptor, and the estradiol receptor will actually give a positive feedback to the oxytocin receptor, and then oxytocin come from the pituitary, and then the CL from the ovary. Uh, this will actually increase the prostaglandin synthesis, and then prostaglandin synthesis actually will lead to the production of PGF2 alpha hormone. And then this PGF2 alpha hormone will go to the CL and will cause the luteolysis. And then there will be luteolysis, there will be a no progesterone, progesterone will go down, and animal will come to the heat. So what if the cow is pregnant? That question comes again. What happened? That was the normal process. So if the animal is pregnant, then the conceptus or embryo will try to save himself. He will send a signal called interferon tau. That interferon tau will go to attach to the uterus to the interferon receptor, and then this interferon, uh, after attaching with the receptor, will actually block the estrogen receptor and then the oxytocin receptor, and then eventually will decrease the PGF alpha production, and there will be no loss of progesterone. So progesterone CL will remain there, progesterone will remain there, and that's how the embryo actually rescue the CL and pregnancy will maintain. This is called the maternal recognition of pregnancy. And you can access this paper through this QR code. This is adapted from this paper. And this is actually first school of thought. Most of the people believe is this is the maternal recognition of pregnancy is happening, but you will see there are some other school of thought as well. So we talk about the second school of thought. Second school of thought is almost the same. Like when the concept is got elongated, release the interferon tau, and it will goes. I will not go into the detail. It's the same with the nucleus is producing basically will block the oxytocin receptor and estrogen receptor. But what is important? There is a third another point. There is a prostaglandin synthesis inhibitor, production of synthesis inhibitor. So you will see here the arachidonic acids go to the prostaglandin synthesis, we will produce the prostaglandin, and then arachidonic acid will also convert into epoxygenase and epoxygenate derivative. That they all of them is actually increasing this prostaglandin inhibitors. So when there is more prostaglandin inhibitor, there will be a no production of prostaglandin. So this is called the second school of thought. They say instead of interferon tau blocking the oxytocin receptor or estrogen receptor, is actually blocking the production of prostaglandin. This is called the second school of thought. So if we go actually in the biochemistry of this prostaglandin production, so actually the all of the prostaglandin, their precursor is the same. It's called arachidonic acid is a primary precursor. there is a backward acid is actually converted to the through different enzymes, COX-1 and COX-2 convert into the PGH2. This PGH2 hormone is the intermediate product is actually convert into PGF2 alpha. I'll convert into PGHE2, and there is another prostaglandin is PGE1, and there are a lot of other as well. So the main point is the PGH2, if they are different enzymes, they will be converted into PGF alpha, and the same PGH2 will be converted into PGE2. And we will see in the next slide how the PGE2 is important. Now, the role of PGE2 for the maternal recognition of pregnancy, if we see the interferon TAR, there was a study back in 80s suggesting if we gave the PGE2 to the non-pregnant Jews, it extends their estrous cycle. That means it's kind of saving their corpus luteum. So PGE2 is somehow luteoprotective, like is saving the CL. And the Ruminant, another study, they describe that the PGF-alpha is a luteolytic hormone, like it will destroy the CL, corpus luteum. But the PGE2, in contrast, is a luteoprotective. It will protect the CL. And a very important thing to remember here is interferon peaks at day 23 and dramatically drops after day 26. So the main point is there is no interferon in sacramental gestation. Why it is important? Because I'm discussing in the further slide, and you will see how it is important. So by keeping in these thing in mind, there comes a third school of school of thought, and the third school of thought they think that intrauterine prostaglandin biosynthesis and signaling are selected directed toward PGF2 alpha during the luteolysis and toward PGE2 during the establishment of pregnancy, the sheep. That they actually prove that if there is animal is open, then the PGE2 machinery, everything is worked to convert the PGE2, PGH2, which is intermediate product, to the PGF2 alpha. If the animal is pregnant, basically that PGH2 will go to produce the PGE2. So it's not like interferon TAR, just PGF alpha is not producing, and there is a PGE2 which is producing, and that PGE2 is actually more luteoprotective as well. So they actually proved this by designing a study. They selected some sheep, and they at day 12, 14, 16, four per groups per day, and they randomly randomized them, and they took their all kind of blood sample and corpus luteum, uterus, and OU2 ovarian blood sample as well. And then they see the interluteal concentration of PGF-alpha and PGE2, and you see at day 16 in cyclic cow, PGF-alpha is higher. Cyclic means non-pregnant, and the PGE2 is lower, but that machinery is shifted at at day 16 in pregnant, PX represent the pregnant animal, and then in pregnant animal, the PGF-alpha is lower and PGE2 higher. Then mean the machinery is just shifting to the PGE2. So right now we don't know what are the actual factors. Some say in the interferon TAR is this through just blocking the oxytocin and estrogen receptors. Some others say they are just blocking the production of prostaglandin, and some others say they're just the PGE2. And some also believe that it's actually the interferon TAR which stimulates the projection of PGE2 rather than anything else. But still, nevertheless, whatever the actual pregnancy recognition factors, still the CL cannot be rescued due to impaired maternal contraction that lead to pregnancy loss. So anything happened to the CL because something is problem with these factors will lead to the pregnancy loss. There's actually in second month of gestation, they observe that the conceptors which are like conceptor demise, all kind of embryo loss, 60% are because of the CL regression. And they also observed in lactating dairy cow, in all the AI, 16% of pregnancy loss because of regression of CL. In IVF embryo 25%. And in the case of clone, that number reached up to level of 80% of pregnancy loss. So these things keeping in mind, here comes another proposal. They propose another model. The group of study, the group of people from our lab, they study this design. They actually what they did, they actually synchronize all the animal, and on day six, on day five, they give them GnRH and they give ultrasound as well, and then they give they they perform a ultrasonography at a regular interval up to day 75, and did a back check as well, and they took their pregnancy test as well. And they actually given the GnRH mean they are creating accessory CL and like one extra CL. If the CL, this is the actual CL. If the new CL or accessory CL is on the other side of the ovary, it's called contralateral CL. If the new CL is on the same side of the ovary where actual original CL was present, it's called ipsilateral CL. And they observed that almost 60%, 65 in primiparous, in 67% in multiparous, contralateral CL regression happened. And further on, after day 33 onward, they observed almost 85% in primiparous, almost 60% contralateral CL regression happening. What this suggests that after day 33, if more than 66% of contralateral CL regression happening and also ipsilateral CL also regressing, this suggests that there come another pulse of prostaglandin PGF2 alpha, which is causing the luteolysis of the CL. And there are some factors which are saving the ipsilateral side of CL, but they are not able to save the contralateral side of CL. So what is happening? We already know like the interferon Ta is not available after second month of gestation. So we are assuming that there is a second critical period. Like the first critical period was in first month of gestation and during the day 16. And there is a second critical period in second month of gestation. And we don't know at what days exactly. But there is a second critical period and there is a PGF-12 pulse is coming, and there are some other factors that need to be determined till now, which are just saving those ipsilateral CR. So now the question arises: how do we determine whether the pregnancy lasts? Is due to the corpus luteum regression or the embryo demise? Because if any pregnancy loss happen, we don't know. This is the fault of embryo because there was something wrong with the placenta. Maybe there's something wrong with the embryo and embryo was genetic defect or something like that. Or maybe because the CL regress and that's why we are losing the pregnancy. So our group at Wisconsin, they studied this thing and they realized they actually do hormonal analysis, two different hormones, progesterone and PAG, and they determine progesterone and PAG, if they go together, PAG is actually pregnancy-associated glycoprotein, which is released from the placenta after the attachment. Usually placenta attach around day 19 onwards. So you see after day 22, the PAG are going up and the progesterone is already up. So if they both are okay and they both are going at the same level, that means there is no pregnancy loss. The animal is just pregnant. But if you see here, luteolysis before the decrease in PAG. So you see progesterone is decreased before, but the PAG remains still there for 2 or 3 days. That means the placenta was intact and the PAG kept releasing. But the progesterone goes down, that suggests the CL get regressed first. And that pregnancy loss is due to because of CL regression. And in the second case, you'll see the PAG drops, but the progesterone was still there. That means the CL was there, but this PAG drop is just because something happened with the embryo. So that's how they determine this stuff. By keeping in mind these kind of study, and we propose development of model with the Hue for the identification of second signal of maternal pregnancy in ruminants. That's a study what I am doing. We synchronize the sheep. I'm doing the three things: the blood sample, also the IST sample, and also the ultrasound sample. So IST is interferon stimulating gene. Basically, we want to check how early we can go and detect the animal was pregnant but lost the embryo. And we, after day 10, we are just dividing them into these four groups, and we're just keeping with a single CL and double CL. And then again, after day 10, after every 5 days, I'm doing the ultrasound till day 60. I'm taking a blood sample every day. And I'm taking after till day 20, after every 2 day interferon stimulating genes. And then after 5 days, up to day 40, interferon stimulating genes. Uh, that's how we are just categorizing our different groups. Non-bred, non-pregnant sheep, bred, but non-pregnant sheep at day 60 is 17 and 60, bred and pregnant sheep at day 17 and 60, bred and pregnant sheep at day 17, but not at day 60. And also the bred pregnant at day 17, but not at 60. One is due to the loss of CL recession, and one is due to the loss of embryo demise. And then I would just acknowledge my parent university, the Cholistan University of Veterinary Animal Sciences, Bahawalpur, and then the Higher Education Commission, my advisor, and my group member. Thank you.